All right. Once again, thanks everyone for being here today. I'm really excited to tell you about my work that I have planned. And yeah, I'm going to be talking about grape powdery mildew and botrytis, and then some of the ways that we manage these diseases, and then hopefully we'll be able to mitigate fungicide resistance in the future. So to start off, U.S. grape production is a huge part of our economy. The fruit itself has a large value. The farm gate value for grapes in the U.S. is $6.6 billion. But not only the value of the fruit provides to the economy, but also the people that it brings into the vineyards. And agritourism actually provides about $18 billion to the U.S. economy. So these are a very important crop to the U.S. economy. But one of the things about grapes is that it's completely dependent on fungicides. About 95% of yield is actually attributed to fungicide use. So absolutely must use fungicides to get a good yield. And unfortunately, a lot of these novel chemistries cost hundreds of millions of dollars to produce and take over a decade, a very long time to actually bring to market. So with that, fungicide resistance is actually emerging much faster than these chemistries can be produced, which is meaning we're slowly running out of the tools that we can use to control these diseases. And the diseases that we're trying to control is one, powdery mildew. And you can see that here on the leaves and the grapes. This white powdery appearance is actually the pathogen growing. And this pathogen is the Ascomyces erythrithicator, and it is an obligate biotroph, which means that it actually has to live on living plant tissue. It completely depends on that. And even small amounts of powdery mildew can cause big problems. 3 to 5% incidence can lead to significant reduction in quality or even crop rejection. And a lot of the active ingredient of fungicides that we use is targeted to control this disease, powdery mildew, 90% of active ingredient that's applied. And one of the things that's really difficult about powdery mildew is actual efficient characterization. Because you have that reliance on live host tissue, it really slows down the throughput of your resistance testing. The other disease that we try to control is Grape Bunch Rot, which is caused by botrytis. And you can see the botrytis here actually sporulating out of these grapes. And what's interesting about this photo is that you can see a lot of the botrytis is actually sporulating out of these splits in the grape. And these splits are actually caused by powdery mildew. So botrytis is a necrotrophic pathogen, which means it likes to feed on dead or dying tissue. And it's one of the most important pathogens in the world. It causes about $10 to $100 billion in losses worldwide. In botryticide market, which is the fungicides that are targeted for botrytis, grapes account for about 50% of that market. And even areas that don't get really bad botrytis, every once in a while get a periodic, very severe epidemic, even when we're following our most current recommendations. So the fungicides that we use, there's a lot of them. We have a pretty large toolbox as it is right now. And all of these fungicides here are determined by their mode of action. And they have these different codes called FRAC codes. And FRAC stands for Fungicide Resistance Action Committee. So you can see we have a lot of them, and they're all registered for grape. But I'm not going to be focusing on all of them today, because these are our most commonly used ones. And these is sulfur. Sulfur is our most common one, and it has multi-site, and it's very effective still. Our more synthetic fungicides are FRAC3, which are DMIs, which inhibit plasma membrane synthesis in the fungal cells, and then FRAC7 and 11, which are SDHIs and QOIs, which inhibit respiration or ATP production in fungi. So with this heavy use of these fungicides, there are a lot of concerns on our heavy reliance on them. One is consumer demands. The consumers are demanding that we have these really beautiful, high quality fruits. And a lot of our varieties of grape that are more resistant to powdery mildew aren't as in high demand, so there's not really a market for them. There are also some environmental impact concerns that have with fungicides, with things like runoff. And then also health concerns for those who are actually being exposed to these fungicides in the field. And then going back, when we consider the production costs to make these new novel chemistries and the rate of fungicide resistance, this isn't really a sustainable system. And I'm not saying that we can completely cut out fungicides, but we need to be thinking about ways that we can enhance the sustainability of our fungicide use. So I plan on attacking some of these problems with some of my objectives for my dissertation. And the first one is we want to develop a high throughput method to look at obligate biotroph resistance, especially in powdery mildew. And then what we want to look at is sort of how do we make our chemical management of these diseases better? How do we increase the efficacy of them? And then thirdly, what we want to do is we want to look at Botrytis inoculum, its epidemiology. Where is it? Where is it spreading? And how is it resistant to the Botryticides that we have available to us now? So I'm going to start off with our first objective, which is looking at developing a more high throughput assay. So the current standard way to test DMIs, or demethylation inhibitors, which I mentioned earlier, is a live leaf disc. As you can see on the right, there's plates with a bunch of leaf discs, and then there's an actual leaf disc zoomed in that has sporulating powdery mildew. The drawback to this assay is that it's incredibly time and resource expensive. And it doesn't give you great data back because these discs commonly can become contaminated or die, and you're just really kind of guessing how much area of it's sporulating. So we wanted to come up with something better than this that was a little bit more objective. And so the rationale behind this was that powdery mildew spores, when they land on a surface, they begin to germinate. So just that contact causes them to germinate. But because they're obligate biotrophs, they're going to have to actually use the plant to get nutrients. So they have to bring their own things along with them. They have to bring their own water, their own energy, like these different metabolic precursors. But because they need this live host before infection, if they can't get there in time, they're going to run out and they'll have to start making these things from scratch or de novo. So our hypothesis here is that an inefficient synthesis of these sterols will be expressed then as reduced growth of these germlings or these germinational spores. So this assay is built around this idea of that we take a 96-well microtiter plate and we add in fungicide amended gelzan medium, which is very similar to an agar-like medium, but it's much clearer, so the images are much better. And we need that clear imaging because what we're going to do is we take these plates at the bottom of a settling tower and we disperse spores with a little puff of air in this little trash can setup, and the spores will actually distribute on the plate. We image that with this nice digital microscope, and then we'll actually image these same wells 48 hours later. And what we do is we'll take then these images and process them in ImageJ so we can get data out of them. So what we're actually quantifying when we take these images is the amount of Hyphal growth. So we're going to be looking at the change in Hyphal area calculated, and we're going to compare it to the treated and untreated. So on untreated, we have these nice spores with these long germination tubes. But then when we have actual treated spores, you can see the germination tubes are much smaller. And this is really what we're looking at here, is how much of this area has been reduced. And from that, we're actually able to build some dose response curves that you can see here. So we have concentration on the x-axis and then our relative growth on the y. And from this, we're actually able to make our IC50 estimates. And what we found is this has been pretty good agreement with our live leaf disc assays and also our qPCR results, which look at the mutation that's commonly associated with DMIs, DMI resistance, I mean. But what we also found was the kicker to this is that this can test more than just DMIs. We're able to test FRAC7, which is our SDHIs, FRAC11 QIs, and then FRAC13, our quinoxaphin as well. Really what we're able to test is up to 24 different treatments of fungicides and concentration combinations. So you can see that on this is that in our treatments, we have all these different treatments. So these are different fungicides at different concentrations. And then we have our actual relative growth here. And what we found is that it works for these chemistries as well. We're able to compare this to our previous bioassays and then our qPCR results for the genes that are commonly associated with resistance to these fungicide groups as well. So then moving on, to conclude with this, is that this assay is way better than any live leaf disc assays. It takes half as much labor that you need, and we can process 3 times as many isolates at the same time. So it saves us a ton of time and which equals a ton of money. You can turn over these assays and get more of them started much quicker. 2-day incubation for this high throughput assay as opposed to 2 weeks for the live leaf disc assay. And what we've calculated, this comes out to about a savings of about $150 per isolate, which when you have an isolate collection of 100 or more, really adds up. So we've found that this has been a really great addition. So now moving on to objective 2, what I'm going to be talking about is using UVC radiation actually to help us boost our efficacy for the control of mildew and bunch rot. So using UVC light. So here on the right, what you can see is a setup to expose the grape canopy to UVC light. So we plan on doing this, but UVC light is a non-ionizing germicidal radiation between 200 and 300 nanometers. And how this works is that fungal DNA repair genes to UV light are turned off at night because there's no real need. The sun's not out, so you're not having this damage, so they turn them off. So the use of this UVC light is much more effective at night when these genes aren't on and they're more susceptible to it. So what we're hoping this will do is not only control disease, but also reduce our chemical inputs. So our hypothesis with this is that UVC light in conjunction with our fungicides is going to better manage disease and also reduce our fungicide resistant populations, which I'll get to in a second. So how are we going to test this in the field? We have this setup called the Corvallis Dragon, which is of a fleet of dragons around the country. And this is a project in collaboration with David Botrytis at Cornell. And so what this is a tractor-mounted array of UVC lamps. So inside this tent-like structure here, there's a bunch of UVC lamps which will be exposing the canopy to UVC light. So how are we going to use this in the field? This is the experimental design. So we're going to have whole rows in our vineyard that are randomly assigned a UV treatment of either no UV, a low dose, or a high dose. And then we're going to have buffer rows in between to reduce plot-to-plot interference. But not only that, we're going to have some treatments within those rows, so a split plot. And these are going to be 5 vines within a row that are going to have different fungicide treatments. We're going to have sulfur, which is our most common fungicide at 2 different intervals. We're going to have treatments with synthetic fungicides and also sulfur alternated with our synthetic fungicides, and then untreated. And what we're going to be looking at is how powdery mildew disease is in these plots, and then also botrytis, phytotoxicity caused by the UV light, and then our QOI resistance that we see out in the field. But we're also going to be doing some more lab-based things. We're going to be looking at UVC fitness costs for resistant isolates. So what we plan on doing is taking either a powdery mildew culture, which you see here, or a botrytis culture, and we'll expose it to UVC, maybe using a conveyor belt system like this, where we might have it run along and get this nice controlled exposure to UVC light. And then from there, what we'll do is we'll actually assess the fitness of these. So after exposure to UVC light, we'll look at Hyphal growth. How much do these cultures grow after this exposure to UVC? We'll look at conidia or production, or even the viability of conidia after exposure to this radiation. So how we plan on doing that is, once again, using digital imaging on this microscope, but we'll use different software. There's a software called Fungal Feature Tracker, which will help us estimate the amount of Hyphal growth, how many conidia, and then also we can look at viability that way as well. And hopefully what we'll be able to do is then evaluate fitness of these fungicide-resistant isolates. Does having the QOI resistance gene of G143A, is that causing a fitness cost? Is it more susceptible now to light because of this mutation? So that takes me to my third and final objective, which is going to be then looking at botrytis. Where is it? How is it spreading? Where is it spreading to? And then also its resistance. So botrytis inoculum first. So what you're seeing here in the photos is this is some old grape cluster from the previous season. What you can see is actually botrytis is sporulating on this. So any debris that falls off the vine that isn't harvested can be an inoculum source. But not only are we going to be looking at stuff that's coming from the vineyard or the canopy, we're going to be looking at other things in proximity. So this is a rosebud that has very severe botrytis infection and is producing a ton of spores. So botrytis infects many, many plant species. So a lot of different plants that could be in proximity to vineyards could be potential sources of inoculum. And that's what we want to understand. So we're going to be sampling floor debris and landscape plants that are in proximity And what we're going to look at is their sporulation potential. When we inoculate them, how many spores are they producing? Are they going to be significant sources of inoculum during the season? And then what we'll hope to do is actually be able to identify some of these isolates using simple sequence repeat markers to sort of understand and track what might be considered like an individual inoculum source and see where is it and how does it specifically spread throughout a vineyard. So then we also want to do some Botrytis inoculum monitoring in the season. So not just where the source is, but how is it spreading as well. So we'll be sampling at vineyards that have known Botrytis issues, and we'll be sampling clusters themselves to see what is actually on the clusters. But we'll also be taking samples from impaction spore traps. So this will be what we do to monitor the airborne inoculum Botrytis. So we'll be able to see where is the Botrytis in the field during the season on the fruit, and how are these inoculum levels sort of changing throughout the year? Are they peaking at certain times? Are they in lulls at certain times? How are they changing throughout the season? Because that's going to really improve how we can make management decisions and how do we time these things. Moving on, then we're going to also be looking at historical DNA. So our lab has actually been collecting a lot of this DNA for over a decade to sample for powdery mildew in the air. But in theory, there should also be Botrytis on these spore rods that we're collecting powdery mildew. So in theory, we could come back, take samples from this DNA that was used to monitor powdery mildew and actually just monitor Botrytis. So we could use qPCR and quantify the amount of Botrytis conidia in these samples. And then so we have this huge historical record of how Botrytis has been changing over the last decade. And then we can look at the conditions from that year. How was the weather? What was the how much rainfall was there? What was the different control applications? You know, we can take in a bunch of things and look at how it's all fitting together with the amount of Botrytis we see in the field. And then we also want to characterize Botrytis fungicide resistance. So Dr. Stockwell has seen that in berry crops, about 61% of the isolates were resistant to at least one chemistry, one Botryticide chemistry. But within that, at least over or 64% of those were resistant to more than one chemistry, so multi-resistance. So what we want to do is we want to do something similar, but for grapes, because this is well known in berry crops, but maybe not as well for in the vineyard setting. So what's actually on grapes? And so what we're going to be testing is the whole battery of these fungicides that are registered for Botrytis control in grapes. And how we're going to be doing it is, once again, using 96 well plates, but we're going to be using a broth that we're going to amend with fungicides. And then we'll actually be measuring the growth as optical density. So as the Botrytis grows, it'll make the broth cloudier, and then our optical density will begin to increase. So we can actually sort of plot out these growth curves. So that's what you can see here on the graph, is that on the x-axis, we have our time or hours. And then on the y, we have our optical density. And then each color represents a different concentration. So we have this method working for 4 of our frac groups: Benamil, fluopyrium, which is an SDHI, tebuconazole DMI, and then trifloxystrobin, which is a QOI. And this is how we plan on testing fungicide resistance of Botrytis that we find in grapes. We still have to work on this method for the other fungicide classes, but that's something that we're working on right now. And so why is all of this work important? It's really important for us to develop these tools like the assay that I talked about to make more informed management decisions. We can now more quickly assess where is fungicide resistance, which fungicide resistance specifically are we dealing with, and so we can make better decisions there. We're going to be able to then integrate more sustainable options like UVC light to not only increase our efficacy, but then also extend the life of these fungicides so that resistance isn't taking such a toll. And then finally, we're going to be able to improve plant disease management recommendations. If we understand our pathogens epidemiology better and how it's spreading, where it's starting, we can make even better recommendations to control disease in the future. And so with that, I'd like to really thank the Foliar Pathology Lab and everyone that's on my committee: Walt, Chris, Dave, Achala, and Christine. And with that, I'll be happy to take any questions from anyone. Thank you.
All right. Good job. Do we have a, let's see, looking at questions. Melody?
Yeah, nice presentation, Alex. I'm curious about the UVC possibility for commercial application. Do you see growers being able to use that technology should it prove effective?
I think so. I think that it could be used. I think that we'd have to look at how much it's increasing efficacy to see if it would be economically feasible. But I think, you know, the way that we can mount it onto tractors and stuff, like logistically, it's completely possible for growers to use.
I have a question, Alex.
Yeah.
Great talk, by the way. Mine's along the same line as Melody's was. I mean, you probably already have some idea how much UVC dose you need, and if you could maybe explain that a little. And also, just curious, is there any potential for UV damage on the grapes?
Right. Yes. So we're going to be testing at 2 different doses, sort of a lower dose and a higher dose. And yeah, we haven't really, there have been studies that show that the doses that we're working with are pretty similar to previous literature. But yeah, there are also some concerns that there could be some phytotoxicity, and we'll be looking at that. You know, in cucurbits, they were looking at using UVC light, and they did see at some really high doses that there was a reduction in the actual amount of like photosynthetic area. And I think it's also important to note that, you know, for a lot of our smaller growers, this would be very easy to do, you know, something that you could retrofit onto your tractor and just go through your vineyard and apply that. And I think that's who would actually benefit the most, is those who, these small growers who really need that, that efficacy boost.
Thanks.
Susanna?
Yeah, very nice talk. Thank you. Just curious about follow up on Tom's question a little bit. Wondering about what kind of permitting for the UVC you guys need to get, and also any kind of damage to potentially beneficial insects or other wildlife. Right.
So yes, this is quite dangerous radiation. Even short exposure to just yourself could cause some serious burns. So there are a lot of safety issues that we'll have to address. You know, we all have to wear the proper PPE that completely covers, you know, every square inch of us. I am not sure what the damage would be to beneficial insects and such, but that would be definitely something that would be important to look at. I think that there might be some like damage to maybe mites or something, but I think that would need a little bit more study.
Todd? Ooh, Todd, you're breaking up. Can you try that again? Jay?
Say, Alex, do you expect any, since Botrytis is so ubiquitous, do you expect any differences in fungicide resistance from the work that Virginia's done on small fruits versus on grapes?
I would expect it to be pretty similar. I'd say a lot of the, if being that both the berry crops and grapes can be infected by the same Botrytis, you know, it's not outlandish to think that the Botrytis could be moving, you know, from these berry crops into our grape, you know, maybe hopping along some other hosts along the way. So I would expect it to be pretty similar to what we see in berry crops, yeah.
Ricardo?
Yeah, great talks. All the ones that I have listened up to now. I was wondering, are there times of the year, of the season, where some of these methods would be more beneficial if you are really I'm thinking that during wintertime, you don't have so much problem with photosynthesis effects, etc.
Right. We're going to be using this UV from basically our first, during the season, you know, we're going to start at about 6-inch growth in our shoots, probably through veraison, so through August. But we have not done anything with looking at any of the like overwintering structures, like is UVC light actually going to be doing, reducing the amount of like primary inoculum? Is it going to be efficacious against Chasmathesia and things like that?
John?
Hi, Alex. That assay in Objective 1 is really super cool. And sort of wondering, it's really indicating, seems to me, that your fungicides are acting really quickly and sort of affecting things really early in the fungal development, at least in your in vitro system. And I wondered whether you think or have any data to sort of you know, how quickly are those fungicides acting, you know, out in vivo on the plants? Are they affecting, you know, the spores that rapidly, or is it later?
Right. So a lot of the fungicides that we use are actually what we might consider fungistatic. So they don't really kill it; they just prevent the spores from like growing. You can think of it as, you know, as Ken commonly tells me, is like it's like painting a house, right? You're just preventing it from doing anything. Not a lot of our fungicides, some of them do have what might be called kickback activity and actually can knock back the pathogen once it's actually infected. But a lot of these are just actually preventing the germination of the spores. So they have a pretty immediate effect on these fungi, but once they're washed away, they really don't have any effect anymore.
Thank you.
Yep.
David Jent?
Hi, Alex. So azoles are used in clinical settings for treating fungal infections in humans, and azole resistance is a huge problem. Are you familiar, which is the same chemistry as DMIs? Are you familiar with how azole resistance is assessed in a clinical setting? And I guess a follow-up to that would be whether this high throughput assay has any applications in those contexts.
Yes, I have seen some studies that have looked at some of the medical side of azole resistance. And what they do is they basically have what agar, and they put some spore, that's fungicide adjusted, and they put some spores in the middle and they measure radial growth. So no, it's not very high throughput because there's pretty good incubation period. And I remember some of these studies had pretty low like number of replications, maybe only like, you know, one, like in a little like plate with a couple like big wells in there that has fungicide amended medium. So that's at least one of the studies that I remember, but I don't remember it being a very high throughput method at all.
Cool, thank you.
Susanna?
Yeah, I'm curious in your Objective 1, when you're looking under for the spores under microscope, I was wondering if I can't remember if you've already done some of that or if you're planning on doing that. Are you using any software for counting the germinated spores, or is that done all by just counting, or any technical bits you can share about that?
Right. So after we get the images, we process them in ImageJ with a macro called Hyphal tracker. And that allows us to sort of remove the background from these images and then filter out anything that might not be actually a spore. Let's say some like, small pieces of dust get in there. We can actually filter those out. And what we'll be able to do is then calculate the area for each of these. And I get this big output file of each spore or region of interest, as it calls it. It says, you know, all these stats about it, including how much area it has. And that's how we're able to get all this data. I don't do anything by eye. Everything's done by this this program.
Okay, great. That's what I thought, but I just wanted to ask. Thank you.
Yeah.
Any other questions for Alex? All right. Let's unmute and thank Alex. Great talk.
Thank you.
Good job.